papillomaviruses. So papillomaviruses are kind of interesting in terms of how the virus works. So these things actually, unlike everything else that we actually call a virus, they don't actually encode their own polymerase. If I remember correctly, they actually interact with some cellular polymerases and make the cell copy for them. So you've known viruses that make the cell do all the translation for them, and this is very much the next level. So papillomaviruses are well known as viruses that cause things like warts. And this would include warts anywhere on your body, including those little tiny things they call like skin tags that you might get on your eyelid, for example. And they also include genital warts as well. And they also include plantar's warts, so those things that you might have grown on the bottom of your foot. I had a whole bunch of them. We used to go to the YMCA a lot when I was a kid, and I think I must have picked them up there because, yeah, I had to have like 30 of them cut out of my foot. It was terrible, but whatever. You bounce back. All right. So how do these things work? Well, they're actually very interesting. Of course, right? Yeah, these are viruses we're talking about. So let's kick on the little subtitles so we can talk about them. There we go. So papillomaviruses are going to include the viruses that cause cervical cancer. And if you've had the cervical cancer vaccine, and this is offered to men and women, yeah, and probably should be, then you have had a vaccine against this particular virus, which is kind of neat. So part of the course is normally that we do a vaccine debate where we would talk about, I would have you guys take sides on the science and the way that that's going to be done. What we're going to do here right now is look at just some of the basics of how the virus works. All right, so there's our little papillomavirus, human papillomavirus. And there are hundreds of different strains of these things. We break them down into different groups and we tend to give them a number. And there are certain number papillomaviruses that will be associated more with cervical cancer or more with throat cancer. For example, Michael Douglas had a papillomavirus cancer. And they tend to have common causes, or rather, you can catch the same virus from a variety of different ways. So what the virus is looking for in a nice host is it would like to get down to your basal cells right here, which are the lowest layer of the epithelium. So on the outside, you're going to have the cornified epithelium. These are cells that are either dying or dead, the ones that are kind of dehydrating, and these are just forming a barrier layer. Then you got your granular layer, they're on the way to becoming those. You got your spinous layer, and you got your basal layer down here. And it's been a while, but if I remember correctly, I think the basal layer gives rise to all the other layers, and they just keep going up and then sort of sloughing off, getting scraped off on the inside. So the virus needs to get right down to the bottom. And so the question is, how can the virus get there? How does it get down underneath, a couple layers of your skin? This is where various kinds of human activity come into play. So, for example, this is why this is sometimes a sexually transmitted disease. The sex has nothing to do with it, but what the sex will do is actually damage the epithelial layer. You'll have occasional little areas where you've rubbed away too many of these cells and where the papillomavirus can potentially get access. And it doesn't need a giant canyon like that to get in. It can worm its way between cells if they start to get loosely attached. Anyway, once it gets in there, it's actually going to grow in these cells in the basal layer. And as it matures, as the cells mature, rather, the virus also matures. So it's going to get into these bottom cells, and then they're going to make these cells that make these cells, make these, until it finally releases. And you'll have different programs of gene expression. Some things that are turned on in the bottom basal cell, some things that are turned on after it's divided and become one of those other cells in the spinous or granular layer, for example. And then finally, the virus is going to be released when it gets up to the top. So once again, if you have a lot of contact with these top cells, the virus has already done all of its replication in sort of this area, and it already infected these cells at the bottom. And once the virus gets in, it's going to turn these cells cancerous. So papillomaviruses cause cancers, and that's what every wart you've ever had actually is. It's a very small, localized cancer. Yeah, plantar warts on your foot, little teeny weeny cancers. They're not cancers that are going to spread throughout your body. They don't usually metastasize or anything like that. But yeah, it is a basal cell cancer of a sort induced by a virus. So there you go. All right. Oh yeah, and we should go back. So the genes of the virus are divided up into early genes, which have an E at the front, and late genes, which have an L at the front. And so the early genes are going to be responsible for taking over the cell and getting this virus copied. Then the late genes are going to be responsible for making the brand new virus and packaging it in there and getting it out of the cell. And so if I remember right, the L1 in particular is the thing that they usually use for vaccines when they're vaccinating. Okay. Here, so we're going to go through, these are from a review, and I actually posted the review on Blackboard, so you can go through it, you can have a look, you can get a better sense of what's going on. We're going to start with the big overview. Then we're going to go into the details. So you're going to see things on here and say, well, that's not exactly what I see on the next slide. This is the big picture. We're going to get into the details, but this is the overview of what happens. That's the best I can tell you. All right, so there's going to be 3 key proteins from the virus, and they're 3 of the early genes that make those proteins. So it's early gene 5, E5, and early gene 6, E6, and early gene 7, E7. And so what they do, E5 does a number of things. And these things are going to contribute to E7 and E6 working a little bit better. So that's what the dotted line up here means. E6, interacts with these proteins, or E5 interacts with these proteins and interacts with other proteins in the cell in such a way as to make E7 and E6 a little more potent, a little more powerful. So E7 is going to be aimed at driving aberrant proliferation, which means the cell's going to keep dividing. And if you get a cell with this in there, it's basically trying to push down the gas pedal. If the gas pedal breaks and it's stuck on and there's no more cell cycle control because E7 has done its job, then you're going to get a process called immortalization, which is a fancy way of saying that cell is going to become cancerous. Cancer being a cell that just keeps dividing and doesn't ever really stop dividing. So E6 is going to come at this from the other side. So E6 is actually going to stop apoptosis, and it's going to stop cells that are trying to stop proliferating. So E7's pushing on the gas, and E6 is cutting the brake lines. I think that's a nice way to look at it. And E5 is, like a little mastermind that helps them both out doing their jobs. And so E6 can also lead to immortalization. So either one of these separately, E6 and 7, can cause immortalization, cancer formation. But together, they're real good at causing cancer formation. And so, this is a process that we call transformation, as in they're transformed to cancer. That can just continue to grow. And that is bad. All right, so going through these proteins one at a time, I think we're going to do 7, then 5, then 6. I think that's the order, but we'll see what order the slides come up. Yeah, who knows, and I'm not going to look ahead. So we'll start with 7, because 7 is kind of all over the place, but there's a couple important things that it does. So E7 is going to activate. Whenever you see an arrow, this means activating, like turning on. And whenever you see an arrow, a line that goes out and looks like, talk to the hand, that means stopping. So E7 is going to activate P600, which blocks apoptosis. Neat. E7 is also going to activate IRF1, that's interferon response factor number 1, and P48, both of which will block apoptosis. And E7 is very much going to activate P53, and its job is to, the way that it is interacting with it, it is going to block apoptosis, yeah. And so because apoptosis is blocked, then we're going to lead toward proliferation, yeah. And E7 is also going to interact with a couple other parts of that. So there's a little thing I'll show you in a minute, but it's going to involve P53 and P21. and the cyclins and cyclin-dependent kinases. And together, these are the key and the car, effectively, that is going to cause cancer. All right, and then there's various other things over here. E7 is also going to inhibit a protein called protein RB. It's called the retinoblastoma protein. And that is another way that it is going to actually drive proliferation. RB would try and stop that process, basically. Okay, so here we go. So that's E7's deal in a nutshell. It does a variety of things, and yeah, I would encourage you to look up some of these proteins, like histone deacetylases. What's their role in cell division? Once you look it up, it'll make a whole lot of sense. There are certain histones that are going to be acetylated, and that will either drive or stop cell proliferation, depending on where they acetylate and or deacetylate. And so E7 is interacting with particular ones, and the net result is going to be that we're going to drive proliferation in all these cases. So the cell is going to keep dividing because E6 is making it divide. And these are the many ways how. Sorry, that was E7, not E6. Silly me. Okay. E6, let's come back to E6. And looks like I didn't put in E5. Well, okay. So E5, let me just tell you about it before we go to E6. E5 is going to do two things that tend to make cancer a little bit more likely. One of those things is that E5 is going to cause cells not to display proteins on MHC class 1. So in a human, it would make your HLA A, B, and C not display anything. It's going to inhibit that entire process, which means that you're going to have cells that are no longer showing that they're making proteins they shouldn't be making, like cancer proteins. So a cell like a natural killer cell could theoretically come in there, if it could actually get to them, and find them and knock them out. But the lack of signaling buys the virus some time, and so it tends to work in the virus's favor. The other thing it does is E5 is going to stop the cell. So the cell is normally recycling a protein called the growth factor receptor. Let's just call it the growth factor receptor. And so it'll go up to the surface, then it'll come back inside, and it'll go up to the surface. And depending on how much is at the surface, how much is inside, that's how much growth factor the cell can receive. And if your growth factor is mostly inside, then it doesn't matter. You're not going to grow. And if it's mostly outside, then that is going to lead to more signals that drive cell growth. And so E5 just stops the entire recycling process. And so your growth factor receptors sit out on the membrane and don't ever get pulled back inside and recycled. And so you tend to get more growth factor activity, which leads to more cell division. Because the virus is going to be linked to the first cell that it infects. And the virus isn't actually going to really spread until it gets all the way into the top layer. And then for the most part, it's going to spread to other people. It's not really going to spread. It's not really going to try to grow. It doesn't have a good way to do that within the cell layer, particularly. So the virus is actually going to copy itself along with the cell, because this is a DNA virus. And so as the cell divides, it's actually going to make new copies of the virus. And so the new cells will have new virus DNA inside them. And on and on and on. The more the virus can cause that cell to divide, the more copies it gets to make. Yeah. And viruses always like to make lots and lots of copies of themselves. And so this is, a real perverse incentive. This virus, if it can cause something like cancer, or at least extremely high rate of cell division, then the virus wins, and it can make more copies of itself. So that's neat. Okay, so E6 does a lot of things, and we're going to unpack one of these things, but I just want to show you all of these little processes. So E6 is going to activate P53. E6 is going to associate with a thing called the E6 associated protein, E6AP, and P53, and it is going to ubiquitinate P53 so that it disappears. Yeah. So E6 is going to affect P53 in such a way that it can't stop cell division. And it's also going to mark P53 for destruction. And so, yeah, one way or the other, E6 is going to make sure that that cell really wants to proliferate and get immortalized. And so E6 tends to be the most powerful gene in terms of the single gene that does the most to actually drive cell cancer, essentially, unchecked cell division. Okay, so let's look at this P53 pathway, because I think it's kind of neat. So you've got your cell, and every once in a while your cell is going to get damaged DNA. And when you get DNA damage, you're going to make a protein called P53. And P53, its single job is to stop the cell from copying itself until the damage is repaired. And so while the repair enzymes are working on that DNA and fixing it back up, P53 is going to be made, and it's just like that little, worker in a hard hat walking around saying, hold on, everybody, hold on, just a minute. We just need to finish something, and then we can go back to cell division. So P53's job is to block cell division. And P53, if I remember correctly, can get activated. I believe it is a protein that can be phosphorylated, but you're going to have to look that up, and the reference will be correct because I'm not remembering fully on that. But P53's job is that it's going to activate a protein called P21. And when P21 is active, it's actually going to be like a switch that binds to a thing called the E-type cyclin E, and the cyclin-dependent kinase. So cyclin is a protein that's like a key, and the cyclin operates the cyclin-dependent kinase. Cyclin-dependent kinase is actually going to turn on a whole bunch of different pathways that together are going to lead towards cell division. So this is the big reason why cells don't divide. Until cyclin E and cyclin-dependent kinase number 2 get together, there is just no cell division in this particular type of cell. This is just how it's controlled. So normally, you would have P53, and it would be activating P21, and P21 would be saying, let's not divide by blocking cyclin-dependent kinase 2 and cyclin E. But if you have turned off P53, or if you have low amounts of P53, then P21 doesn't get activated. And so if P21 is not activated, then cyclin type E and cyclin-dependent kinase are fully active. And once these guys boot up, they will actually inhibit P21 and make it hard to turn P21 back on again until you make a bunch more P53 and activate it, and then you can actually turn it off again. And so this is what the virus wants. It wants to make either low amounts of P53, or it just wants P53 to disappear. Because if there's no P53, then there's nobody to turn on P21. And if there's nobody to turn on P21, there's nobody to stop cyclin and cyclin-dependent kinase from causing the cell to go through the cell cycle and divide. That's the idea. That's the whole idea. So arrest, it would be stop, let's not divide, like when there's DNA damage. And proliferating would be, okay, let's divide again. And so escape from arrest is what the virus is trying to get. And this is how it does it. Just like that. So P53 is something that is involved in a lot of kinds of cancer. You would have seen it perhaps if you were studying skin cancer, for example, or really any type of cancer. It's such a fundamental regulator. When DNA gets damaged, this is the thing that you definitely want to have turned on so that it can prevent your cell from dividing and copying damaged DNA. Because, you can fix DNA as long as you're not trying to copy it and make new cells out of it. Okay, so what does P53 do? Well, it does a couple things. Here is one of the things. What does E6 do? So E6 is down here. Remember, this is the virus protein E6. It binds to a protein that's inside the cell already. And we call this the E6 associated protein because that's how it was discovered. But it's not actually a virus protein. It is actually a regular protein from the cell. And it's a thing called an E3 ubiquitin ligase. And the E3 here doesn't mean like it's early gene number 3. They're just 3 steps of adding ubiquitin, and they're called E1, E2, and E3. So E3 is doing the last step, if I remember right. And it's like a little adapter, basically. So anyway, so let's just call it the E6 associated protein because that'll be a little easier to remember because it's binding E6, and what else could you call it, right? So when E6 binds to the E6 associated protein, it forms a bridge. E6 actually will stick to P53 and the E6 associated protein at the same time. Ordinarily, the E6 associated protein would not bind to P53, but in this case, it does. It really does. And so, as a result, when you have one of these E3 ubiquitin ligases, like this big stripy guy right here, E6 associated protein, it is the last step that says, okay, it sort of marks other proteins for ubiquitination. That's its job. And so it's marking P53 for ubiquitination. Now, there are different kinds of ubiquitination. You can add ubiquitin via different residues. So we talked about a lysine 63 type ubiquitination. And you can add a whole chain of ubiquitins, or you can just add 1 or 2 ubiquitins. And there are actually different results depending on exactly how many you add and exactly where you add them. Ubiquitin is quite a complex molecule when you really get into it. But the main role of ubiquitin, the main reason why we know it, is that ubiquitin is a marker for destruction. So when you add a chain of ubiquitins onto P53, just like that, starting with the first one, then P53 is going to be marked for recycling. And so it's going to go to a thing called the proteasome, which is like a great big garbage disposal. It's just going to shred the P53 into little tiny peptides here and then recycle them. And it's going to use ATP to do this. It's a shredding machine, like a paper shredder maybe. And so this is how the virus wins. E6 is going to bind to the E6 associated protein, and that complex, E6 associated protein plus E6, is going to bind to P53, which is going to call in the other 2 parts of the ubiquitin ligase system. So ubiquitins are going to be attached to the E1 ubiquitin ligase. It's going to transfer ubiquitin to the E2 ubiquitin ligase, and E2 is then going to transfer it to whoever the E3 ubiquitin ligase is holding on to, and so in this case, P53. And once you've transferred one, then other ubiquitin ligases are going to transfer more onto the top of it, and they can attach them at different places, which is how you can get branchy ubiquitin chains. And yeah, there's a particular type of chain that's going to be attached to P53 that's going to lead to P53 destruction. And you remember that P53 is all about controlling cell cycle. And if we get rid of P53, we get rid of P21. And the cyclin and the cyclin-dependent kinase are allowed to, cause that cell to divide at will. So that's the bit that I want you to pay attention to. This is going to be the text question bit. I'm going to have you tell me some things that either E6 or E7 does in order to cause cell cycle escape, basically, uncontrolled proliferation of the cell. And so this is going to be a starting point. And the paper that is on Blackboard is going to be a really nice follow-up to this. I think that's going to help you out. So there we go. That's the end.